Can one of you let me know if my screen is visible and if you can hear my voice?
Yes, ma'am.
Okay. Thank you. So, good evening, everyone. Today is the second live session in this course, Biochemistry, which is taken by course instructor Professor K. Subramaniam. So, in these live sessions, we basically discuss some of the previous year assignment questions, and we not only discuss the answers, but we also try to discuss the concepts behind those questions, so it's easier for us to solve similar type of questions. Okay, so today is a second live session, and we'll be discussing some problems today. And so let's move on to the first question for today. So this is the first question for today. What are the oxidation states of copper Cu in Felling's reagent before and after reaction with acetone? So we have four options: 1 and 2, 3 and 2, 2 and 2, 2 and 1. Would anybody like to try? Anybody? One and two. Okay. Anybody else? So we'll discuss a little bit about Felling's reagent and what it reacts with, which macromolecule presence does it indicate. So the answer will be 2 and 2, but we'll discuss a little bit about Felling's reagent. Then anyone can let me know if you have any doubts. So the theory, this test, Felling's test, is basically given by reducing sugars. Okay, so to the aqueous solution of carbohydrate, Felling's solution is added and then heated in a water bath, and formation of a red precipitate will confirm the presence of reducing sugars. Okay, the copper ions present in the Felling solution is in +3 state, is reduced to +2 oxidation state, and in alkaline medium it is precipitated as red cuprous oxide. So basically, a reducing sugar is any sugar that is capable of acting as a reducing agent. Okay, so in an alkaline solution, a reducing sugar forms some aldehyde or ketone, which allows it to act as a reducing agent. For example, in also Benedict's reagent. So in such a reaction, the sugar becomes a carboxylic acid. So in general, all monosaccharides are reducing sugars, along with some disaccharides, some oligosaccharides, and some polysaccharides. Now the monosaccharides can further be divided into two groups: the aldoses, which have the aldehyde group, and the ketoses, which have a ketone group. Okay, so ketoses must first automerize to aldoses before they can act as reducing sugars. Okay, so the common dietary monosaccharides such as glucose, galactose, fructose, these are all reducing sugars. Okay, so disaccharides are formed from two monosaccharides. We know because di means two. So two monosaccharides join to form disaccharides, and they can be classified as either reducing or non-reducing. Non-reducing disaccharides like sucrose and trehalose have glycosidic bonds between the anomeric carbons and thus cannot convert to an open chain form with an aldehyde group, so they are stuck in the cyclic form. However, lactose, maltose, these are reducing disaccharides, okay, and they have only one of the two anomeric carbons involved in the glycosidic bond, while the other is free and can convert to an open chain form with an aldehyde group. So example, this is the Felling's reagent. The solution A in the Felling's reagent contains 7% copper sulfate, CSO4, and solution B contains 25% KOH or potassium hydroxide and 35% sodium potassium tartrate. Okay, so when the two solutions are mixed in equal amounts, a clear blue solution results from the tartrate forms a soluble complex with the copper hydroxide produced. Okay, copper will react with KOH to form copper hydroxide, and the tartrate basically forms a soluble complex with the copper hydroxide produced. This solution is widely used as an oxidizing agent, preferably in quantitative sugar determinations. Okay, so you can see this is once you mix the two solutions of Felling's reagent, copper hydroxide is formed. Okay, and then when it interacts with the reducing monosaccharide, these are the products. So basically, the objective is to detect reducing sugar in a given solution. That is what the objective of a Felling's test is. So the principle is quite simple. It is one of the sensitive tests for detection of reducing sugars, and Felling's reagents comprises of two solutions, A and B. And Felling solution A is aqueous copper sulfate, and Felling solution B is alkaline sodium potassium tartarate, Rochelle salt. We also call that Rochelle salt. Now, the sodium potassium tartarate present in the reagent, they act as chelating agent in this reaction. And these two solutions are apparently mixed in equal amount before the test. So, when you heat an aldehyde or a reducing sugar with Felling solution, it gives a reddish brown precipitate. And formation of the red precipitate of cuprous oxide denotes the presence of reducing sugar. So, the cuprous oxide will form a red precipitate. That will tell you that whatever the sample was, it was a reducing sugar. Okay. So, if you can see, this is solution A, this is solution B. Okay. And COH2 is forms copper hydroxide. Solution B is the Rochelle salt, sodium potassium tartarate. Okay. So, it's basically the Felling solution is a deep blue color complex. So, this CuOH2, the copper hydroxide, will react with the Rochelle salt to form a complex. As we read over here also, it basically forms, the tartarate forms a soluble complex with the copper hydroxide produced. Okay, 1 second. Yeah. So, the copper hydroxide will form a soluble complex with the Rochelle salt. Okay, and this is the deep blue color complex. Okay, so on reacting with the reducing sugar, it will form a reddish brown precipitate. So these are the reagents. We also need water bath because we need to heat the sample. Pipettes and obviously test tubes. So we can take sample in a dry test tube. We can add, along with the sample, we can add distilled water in another test tube as control. So basically control, it's negative control. We know that it should not give a reddish brown precipitate with water. So that is our negative control. And then we add both Felling's reagent in both the tubes. And we keep in boiling water bath and look for the development of red precipitate. So you can see over here, a positive Felling's test shows a reddish brown precipitate in glucose fructose. Okay, but it will show a negative Felling's test in case of sucrose, starch. Okay, because sucrose, trehalose, these are non-reducing disaccharides. So it will show a negative Felling's test in case of sucrose and starch. So basically, Felling's solution is a reagent which is used to differentiate between the reducing and the non-reducing sugars to determine whether a carbonyl compound is an aldehyde or a ketone. Okay, so we know about the two different functional groups, aldehyde and ketone. Aldehyde is CHO, ketone will be only C double bond O. So we know that acetone, the question is regarding acetone. When acetone reacts with Felling's reagent, what is the change in the oxidation number of copper? That was the question. So we know the structure of acetone. Acetone is a ketone. It belongs to the ketone functional group. Okay. So as the structure of acetone is CH2, CH2 and CO. Correct. So the acetone belongs to the ketone functional group. It has this structure. Now acetones in general do not respond to the Felling's test because they do not have the hydrogen attached to the carbon oxygen double bond. Okay. So the carbon valency is already fulfilled. 1 second. So we know that in case of an aldehyde, there is an H here also to the C alpha carbon. But in here we know that to the functional group C carbon. But here we know that in case of ketone, there is no hydrogen attached to the carbon oxygen double bond, which is present in aldehydes, so which can undergo oxidation. Hence, it is difficult to oxidize ketones with this solution. So ketones are very less reactive towards oxidation. Acetone forms in the ketone group. Okay. However, we need to be careful with Felling's test as some alpha hydroxy ketones also do give positive tests, as these can undergo tautomerization to get converted to an aldehyde form, which gives a positive test towards the Felling test. So any alpha hydroxy ketones might get tautomerized to an aldehyde form. So basically all the aldehydes should give a positive Felling's test, but the ketones are very less reactive towards oxidation and they don't really give any positive Felling's test. Okay, so that's why this test is used to determine the presence of an aldehyde also. So for example, if you see over here, propanol reacts with the Felling's reagent, forming a brick red precipitate. Okay, which is cuprous oxide. So while acetone cannot react with the Fehling solution, so it remains, the Fehling solution remains a deep transparent blue color. So as we know, the Fehling's copper hydroxide forms a complex with tartarate, the Rochelle salt, which is of deep blue color. And when this entire complex reacts with a reducing sugar, it will give a brick red precipitate. So the Fehling's test is positive for this propanol, but it is not positive for acetone. So acetone does not give a positive test for Fehling's. Okay. So basically that was our question. So our question was what are the oxidation states of Cu? So it will not show a positive test. So that is why the oxidation states of Cu in Fehling's reagent before and after reaction with acetone will be 2 and 2 only. Okay, because it will not give a positive test. Okay, moving on to the next question for today. Which of the letters are used to signify the 4 nucleotides found in DNA? Yeah. So it is ATGC, adenine, thymine, guanine, and cytosine. So we have talked about DNA in the previous class. Again, for briefly, DNA stands for deoxyribonucleic acid. And obviously, this chemical substance is very stable. It is present in the nucleus of all cells in all living organisms because this is the hereditary material. It basically transfers genetic information from through generations, and it also controls all the chemical changes which takes place inside the cell. So whatever information your DNA is encoding, that will give rise to the complementary mRNA, and basically DNA will give rise to the mRNA via the process of transcription, and then mRNA will form protein by the process of translation. So if there is any kind of mutation in your DNA, that will harm your protein formation. And obviously proteins handle every function, almost most of the functions of the body, be it structural, enzymatic, most of the functions of the body are handled by proteins. So if your DNA has any issue, obviously that will be translated to the proteins. So it's obviously a very large molecule made of a long chain of nucleotides, and each nucleotide is made of a sugar called deoxyribose, a phosphate group, and an organic base. And the organic bases are adenine, thymine, cytosine, guanine. So ATGC. We don't have uracil in DNA. Uracil is seen in RNA in place of thymine, but in DNA we see ATGC. So these are the 4-letter codes, basically 4-letter abbreviations of these nucleotides, bases basically. So if you can see, this is deoxyribose. It is attached to the phosphate and then adenine. Okay, and they combine to form a nucleotide, and a molecule of DNA is obviously a polymer of all these nucleotides. It's formed by millions of nucleotides joined together by a long chain. So this is the sugar phosphate backbone, and we have the bases. Okay, and DNA consists of a double strand of nucleotides. The sugar phosphate chains are on the outside, and the strands are held together by bonds between these bases. Okay, so this is a double stranded DNA, if you can see. And we know that these strands are complementary to each other. So if we have A over here, this should be T, thymine. If we have G over here, this should be C. And there are 2 hydrogen bonds between AT and 3 hydrogen bonds between G and C. Okay, they always pair up in the same way, as was also deciphered from Shargav's rules. We talked about Shargav's rules in the previous class. Adenine will form a bond with thymine, and cytosine will bond with guanine. Okay, so if there is adenine in one strand, it will be thymine in the opposite strand. If it is cytosine in one strand, it will be guanine in the opposite strand. Okay, so the paired strands are coiled into a spiral called the DNA double helix. This is how it looks. This is the sugar phosphate chain, and these are the bases basically protruding out, and the complementary bases interact with each other. So how does the division happen? So before the cell divides, the DNA strands unwind and separate, and each strand makes a new partner by adding the appropriate nucleotides. And the result is that there are now 2 double-stranded DNA molecules in the nucleus, so that when the cell divides, each nucleus contains identical DNA. Okay, so one parent DNA strand is forwarded to the next generation, and that parent strand would have made a complementary strand by itself. So that is what we call semi-conservative mode of replication. Okay. So this happens, the strands separate, and each strand builds up its partners by adding appropriate nucleotides. Correct? We have talked about genetic code as well. The sequence of bases in DNA forms the genetic code. a group of 3 bases, triplet, controls the production of a particular amino acid in the cytoplasm of the cell. Okay? And the different amino acids and the order in which they're joined up determines the sort of the protein being produced. Obviously, so if there is a mismatch of amino acids, or if there is any mutation in one of the bases, so the frame of reference will change and it will give rise to a completely different amino acids. So the amino acids, as we know, they have the common features of all the amino acids is the amino group, the carboxyl group, and obviously the H bond, H group attached to the C alpha carbon. But the R group is different from all the amino acids. So if a charged amino acid was supposed to be there in place, we have an uncharged amino acid that will basically alter the structure of the protein and hence the function of the protein. So any kind of, even if you think if there's a point mutation, that will make a huge change because your entire frame of reference will change. Your frame of reference will change, so the triplets will be changed, and that next triplet might give rise to a completely different amino acid, which will basically make your protein non-functional or not of that efficacy of how it was supposed to be before in normal conditions. So if you see, this is a small imaginary protein molecule showing how a sequence of 5 different amino acids could determine the shape of the and identity of the molecule. So we know that the primary structure of a protein is basically the amino acid sequence. So this amino acid sequence basically can determine the shape and identity of the molecule. We can figure out from here only if it will have normal shape, if it will be able to carry on its normal functions, etc. Okay. So each amino acid is coded by a particular triplet of bases. Okay. So for example, cytosine, adenine, and thymine, they code for valine. Okay. But cytosine, guanine, and adenine, they code for alanine. Okay. So this is the triplet code, and each triplet codes for a specific amino acid. So this will give rise to alanine. The second will give rise to valine, glycine, then arginine, proline, leucine, glycine, all of these. Okay. So the amino acids are joined together in correct sequence to make a protein. So you can see if one of the nucleotides is absent, somehow gets mutated or deleted. For example, if A is not there, then the entire frame of reference will change. The triplet code will be read as CGC. CGC will give rise to a completely different amino acid. So you can check out from the genetic code table. I don't remember exactly, but CGC might not give rise to alanine. It can give. You have to check the genetic code table, but only one nucleotide change can basically change the entire frame of reference and can lead to a completely different set of amino acids being chained together to form a protein. So that will change a lot of things for the protein. Okay. So obviously the proteins build the cell structures. They are also enzymes, and the DNA controls which enzymes are made, and the enzymes determine what reactions take place. So enzymes can either be proteins or even RNA can be enzymes, as we know. But mostly proteins can basically function as enzymes as well. So if you have any problem in the DNA sequence, it will control which enzymes are being made, obviously, and the enzymes will determine which reactions take place. So if the appropriate enzymes are not there, or if they're non-functional, so obviously those reactions will not take place. And as we know, there's a huge cascade of reactions happening in our body. Like there's a pathway of reactions. If one of the enzymes do not function, then the previous substrate will accumulate, and that might lead to a toxic atmosphere inside the cells. So there's a whole lot of things depending on this sequential cascade thing, that each enzyme needs to be properly functional for the entire pathway to happen. And the structures and reactions in the cell determine what sort of cell it is and what its function is, definitely. Okay. So DNA exerts its control through the proteins, through the enzymes, correct? So genes will be a sequence of triplets in the DNA molecule may code for a complete protein, and such a sequence forms a gene. So if we talk about a protein, we'll know the DNA sequence that codes for the protein, that will be called a gene. Okay, and there may be 1,000 or even more bases in one gene. Correct. So obviously the sequence was the one letter codes were ATGC. You're correct. Moving on to the next question for today. Which of these polymers are not naturally found within any kinds of cells? Correct. So we have talked about protein, starch, nucleic acids. We'll probably talk about in more detail as well. But polyethylene is not naturally found in any kinds of cells. Starting up, we know that polyethylene was first produced by a British company, and then Carl Ziegler prepared a high molecular weight polyethylene at room temperature. Uh, but ICI polyethylene, which was prepared by Imperial Chemical Industries, had shorter branch chains and was waxy and easily deformed, whereas Ziegler made polyethylene was hard and rigid and could easily be drawn into fibers. Okay, and ethylene is an interesting molecule because of its double bond. Correct. Ethylene of its double bond is more reactive than ethane. Okay, so we know the structure of ethane, and we know the structure of ethylene as well. So ethylene basically has double bond between two carbons. It's the formula is C2H4, double bonds between two carbons. So because of its double bond, it is more reactive than ethane. Ethane is an alkane. So ethane does not have any double bonds. It's But ethylene is an alkyne. It is alkyne, and that's why it has a double bond structure. And due to double bonded carbons, make the molecule more susceptible to bonding with other molecules. So when approached by another molecule, the double bond in ethylene splits, and the carbons are joined by a single bond, leaving the molecule free to bond with other carbons. So if you have also heard, ethylene is also used in the artificial ripening of fruit. It stimulates the metabolic processes of the fruit by dissolving into the cell membrane, therefore increasing its permeability. One second. Okay, someone was here. Anyway. So if you have not already heard, it's basically a hot topic even in today's time. There's artificial ripening of fruit, which is obviously not healthy for. We should not. We should we won't even get to know, but it's not healthy for humans to consume artificially ripened food. There are fruits. There are a lot of cases of food poisoning as well that has happened because of this. So, in order to keep the market going, ethylene is used in ripening of fruit. Basically, it stimulates the metabolic process of the fruit by dissolving into the cell membrane and increasing its permeability. Okay, so if we talk about the formation and the structure, ethylene bond yields polyethylene. So if you have a polymer of ethylene, that will basically be polyethylene, and crystalline regions lie in alternating fashion with the amorphous regions, which creates scattering of light. Okay, so high density polyethylene. So absence of branching results in a more closely packed structure, more crystalline, higher density, and chemical resistance slightly higher than that of LDP. LDP is low and high density polyethylene. So low density polyethylene is LDP and high density polyethylene is HDPE. So it basically resists alcohols, acids, bases, esters, and aldehydes. You can see the specific gravity melting point, and these are all translucent structures. In case of low density polyethylene, the small amount of branching on the chain gives a more open structure, and it does not react at room temperature. It also resists alcohols, esters, acids, and bases, and limit resistance to aldehydes. So it can be translucent to opaque, and it is apparently used for plastic food or garment bags, spray bottles, plastic lids, etc. So, low density arises from minimal amount of branching in chain, gives a more open structure. It can be translucent to opaque, and it resists all these estrous bases, but limited resistance to aldehydes, not complete resistance to aldehydes, and it does not react at room temperature. It can be translucent to opaque. Okay. So, what are the properties of polyethylene? It's a solvent for fats, oils, and grease. Okay, dissolving occurs slowly. Electrons are tightly trapped in their CC and CH bonds. Okay, which results in an inability of electric current to flow. Okay, also it results in inability for water and ions to penetrate the interior of solid. So it is basically polyethylene is the most commonly used plastic. It is obviously a polymer, and so many kinds of polyethylene are known, with most having the chemical formula C2H4. So outside N. So since it's a polymer, C2H4 is ethylene. So outside of the brackets, it will be N. So it's basically a mixture of similar polymers of ethylene with various values of N. So it can be low density or high density and many variations thereof. As I said, and its properties can be modified further by cross-linking or copolymerization. So all forms are mostly non-toxic as well as chemically resistant, and as a consequence, these structures are used on immense scale. but its widespread use is also problematic because it apparently resists biodegradation. Okay, and like the hydrocarbon, it is PE or polyethylene is white or colorless and also combustible. Okay, so polyethylene was first, as I said, is synthesized by a German chemist. Okay, who prepared it by accident because he was investigating diazomethane. Okay, and then when his colleagues characterized the white waxy substance that he had created, they recognized that it contained long CH2 chains and it was named polymethylene. Okay, and then as I said, ICI Imperial Chemical Industries took over, and they also, upon applying high pressure to a mixture of ethylene and benzaldehyde, they again produced a white waxy material. So all of these were done, and then the landmark breakthrough in the commercial production of polyethylene began with the development of catalysts. Okay, that promoted the polymerization at the mild temperatures and pressures. So polyethylene is a thermoplastic, that is, it becomes soft and malleable when heated and hard and solid when cooled, and it is an addition polymer. So the double bonds allow for splitting and bonding with other molecules. And polyethylene does not biodegrade unless first oxidized. Polyolefins are highly susceptible to photooxidation or oxidation caused by the exposure to light. Hindered amine light stabilizers are then used to prevent photooxidation. Okay. So polyethylene was the answer to the question. It is not naturally found in cells. The other option was polysaccharides. So we have talked a little bit about polysaccharides. Polysaccharide cellulose is basically, cellulose is a kind of a polysaccharide. So one of the examples of polysaccharide is cellulose. It is composed of glucose units that are connected by beta 1,4 glycosidic bonds. Okay. And polysaccharides are mainly formed when the monosaccharides are joined together. So they can include amylose, amylopectin, cellulose, D-glucose, and differ by branching and types of the glucosidic bonds. Starch is another kind of polysaccharide. It's a storage form of glucose in plants found in insoluble granules, rice, sweet potatoes, etc. It is composed of two kinds of polysaccharides, amylose and amylopectin. Okay. So in starch, amylose basically makes up of 20% of the starch, and it consists of 250 to 4000 alpha D glucose molecules connected by alpha 1,4 glycosidic bonds in a continuous fashion. Okay. So polymers, so these are unbranched. If you can see over, polymer chains of amylose are coiled in a helical fashion. In case of amylopectin, it makes up 80% of the starch. It is a branched chain polysaccharide. So the arrows are misplaced. Sorry for that. It is a branched chain polysaccharide. It contains glucose molecules connected by alpha 1,4 and alpha 1,6 glycosidic bonds. The starches are able to hydrolyze easily in water and acid to give smaller saccharides called dextrins, which will then hydrolyze to maltose and finally glucose. Okay. So in our bodies, these complex carbohydrates are digested by the enzymes amylase. So there's an enzyme called amylase in our saliva and maltase in our intestines. So these complex carbohydrates can provide about 50% of our nutritional calories from the glucose obtained in the digestion. Okay, so ultimately starch, amylose, and amylopectin will get degraded to dextrins and maltose and finally glucose. Okay, so they can also fulfill our nutritional requirements. Another animal starch is glycogen. Glycogen is basically a polymer of glucose that is stored in liver and muscle of animals. It's hydrolyzed in our cells at a rate that maintains the blood level of glucose and provides energy between meals. It is similar to amylopectin because we know that amylopectin from starch is also branched. Amylose is not. It is similar to amylopectin but more highly branched. So the glucose units in glycogen are joined by alpha 1,4 glycosidic bonds with branches attached by alpha 1,6 glycosidic bonds that occur every 10 to 15 glucose units. We have talked a little bit about cellulose before. Cellulose is a major structural unit of plant and woods basically, and it's a polysaccharide of glucose units in unbranched chains with beta 1,4 glycosidic bonds. So they cannot really form hydrogen bonds with water, making it insoluble in water, and it's also resistant to hydrolysis than other starches. It gives a rigid structure to the cell walls in wood and fiber. So cellulose cannot be digested by us, okay, because we humans are not capable of breaking down beta 1,4 glycosidic bonds. Cellulose are not digestible by us. Okay, so you can see the cellulose structure over here. So the carbohydrates can be classified as monosaccharides, disaccharides, and basically polysaccharides. Monosaccharides will form glycosidic bonds to form disaccharides, and then polysaccharides. Monosaccharides include glucose, galactose, fructose, and can be drawn in two structures in two ways, either Fischer projection or Howard Fischer projections or the cyclic Howard structures. Okay. Disaccharides can also form reducing sugars like maltose and lactose, and a non-reducing sugar like sucrose or trehalose. Okay, and polysaccharides are polymers of glucose basically, and as I said, they can be cellulose, starch, glycogen, etc. Okay. Our question was which of these, let me go back to the question. Our question was which of these polymers are not naturally found within any kind of cells. Obviously, protein, starch, and nucleic acids can be found. Polyethylene are not naturally found within any kinds of cells. Okay. So, going to the next question for today. What is the normal human body temperature? Anybody? What do we call the? Okay. So so this is the normal human body temperature. Okay, so we generally, when we check if we have fever or not, we generally use thermometers. For that we use the Fahrenheit rating. Celsius, generally we use these terms in labs. Okay, right, if we have air conditioning equipments that we want to change the temperature of, or we have incubators, in that case we generally use Celsius. At least in India, for thermometers I have seen people using Fahrenheit. We say 98, 99, 98.6, 98.5. All of these, this reading 98.56 is equivalent to 37 degrees Celsius. So when we check for fever also, we check if it's 98 around, 98.5 around, then that's normal temperature. We do not have, not really have fever. Okay. So the physiological temperature of human body is 37 degree Celsius or 98.6 degree Fahrenheit. Okay. So some studies have shown that the normal body temperature can actually have a little wider range also, like from 97 degree Fahrenheit to 99 degree Fahrenheit. Okay. A temperature of over 100.4 degree Fahrenheit, which is more than 38 degrees Celsius, most often means you have a fever. Okay, so fever is caused by illness or infection, correct? Now, body temperature is not uniform throughout the day. It normally changes throughout the day, and in adults, it is lowest in the early morning. Correct? So, it's not that we have body temperature. That doesn't mean that it stays same throughout. Also, human body temperature, it depends on your gender, also age, time of day, exertion level, health status, what part of the body the measurement is taken at. Is it state of consciousness, and also emotions. Body temperature is actually kept in the normal range by a homeostatic function known as thermoregulation, in which adjustment of the temperature is triggered by the central nervous system. So, a little bit about thermoregulation. As in other mammals, thermoregulation in humans is an important aspect of homeostasis. Okay, so what happens is that body heat is generated mostly in the deep organs, especially in the liver, brain, heart, and in contraction of the skeletal muscles. So humans have been able to adapt to a great diversity of climates, including hot, humid, and hot, arid. So there are people living in such diverse, extreme conditions in the world, right? So high temperature pose serious stress for the. Yeah. So high temperatures pose such a serious stress for human body, placing it in great danger. For humans, adaptation to varying climatic conditions includes both physiological mechanisms resulting from evolution and also behavioral mechanisms resulting from conscious cultural adaptations. And for heat loss, we know that there are 4 avenues of heat loss: convection, conduction, radiation, and evaporation. So if a skin temperature is greater than that of the surroundings, the body can lose heat by radiation and conduction. But if the temperature of the surroundings is greater than that of the skin, the body actually gains heat by radiation and conduction. So in such conditions, the only means by which the body can rid itself of heat is by evaporation. So when the surrounding temperature is higher than the skin temperature, anything that prevents adequate evaporation will cause the internal body temperature to rise. Okay, so whenever we're doing sports activities, during sports activities, evaporation becomes the main avenue of heat loss. Okay, humidity affects thermoregulation by limiting sweat evaporation and thus heat loss. Okay, but humans cannot survive prolonged exposure to a wet bulb temperature above 35 95 degree Fahrenheit basically. Uh, Now, what is the control system? So, the core temperature of a human is regulated and stabilized primarily by the hypothalamus. Okay, it's a region of the brain linking the endocrine system. That's a hormone. Endocrine system related. Basically, endocrine system is a messenger system in an organism comprising feedback loops of hormones that are released by the internal glands directly into the circulatory system. Okay, so this hypothalamus is the region of the brain that links the endocrine system and the nervous and the nervous system, and more specifically by the anterior hypothalamic nucleus and the adjacent preoptic area regions of the hypothalamus. So, as core temperature varies from the set point, endocrine production initiates control mechanisms to increase or decrease energy production or dissipation as needed to return the temperature to its set point. Okay. So, suppose in hot conditions, what happens? The eccrine sweat glands under the skin, basically they will secrete sweat. We know that sweat is a fluid containing mostly water with some dissolved ions. So basically it will travel up the sweat duct through the sweat pore and onto the surface of the skin. This causes heat loss via evaporative cooling. Okay, but however a lot of essential water is lost. Okay, so whenever you are sweating, we also sometimes get a lot thirsty when in hot humid conditions. So the hair on our skin, they lie flat, preventing heat from being trapped by the layer of still air between the hair, and this is caused by tiny muscles under the surface of the skin. So relaxing so that the attached hair follicles are not erect. Okay. Also, arteriolar vasodilation occurs. So the smooth muscle walls of the arterioles relax, allowing increased blood flow through the artery. So this redirects blood into the superficial capillaries in the skin, increasing heat loss by convection and conduction. Okay. So in general, we have seen that humans appear physiologically quite well adapted to hot, dry conditions because people are living in deserts as well. However, effective thermoregulation is reduced in the hot, humid environments, such as, for example, the Red Sea, Persian Gulf, where moderately hot summer temperatures are accompanied by unusually high vapor pressures, tropical environments, and deep mines where the atmosphere can be water saturated. So, in hot, humid conditions, clothing can impede efficient evaporation. So, in such environments, it helps to wear light clothing, such as cotton, that is pervious to sweat but impervious to radiant heat from the sun. So this minimizes the gaining of radiant heat while allowing as much evaporation to occur as the environment will allow. Okay, so clothing such as plastic fabrics that are impermeable to sweat and thus do not facilitate heat loss through evaporation can actually contribute to heat stress. Okay, what happens in cold conditions? In cold conditions, heat is lost mainly through the hands and feet. So sweat production is decreased. Okay, the minute muscles under the surface of the skin called the erector pili or pili muscles, basically attached to an individual hair follicle, they contract, lifting the hair follicle upright, and this makes the hair stand on end, which acts as an insulating layer trapping the heat. So we say that we are getting goosebumps, right? Which we can see that the hair on our skin are standing up erect. So this is what also causes goosebumps, since humans do not have very much hair and the contracted muscles can be easily seen. On the inside, arterioles carrying blood to superficial capillaries under the surface of the skin can shrink or constrict, and thereby rerouting the blood away from the skin and towards the warmer core of the body. So this prevents blood from losing heat to the surroundings and also prevents the core temperature dropping further. So this process is called as vasoconstriction. So it is impossible to prevent all heat loss from the blood, only to reduce it. So in extremely cold conditions, excessive vasoconstriction leads to numbness and even pale skin. So if any of you have lived in very cold temperatures, you will see that some parts of your body become numb and pale skin. You can see pale skin. So frostbite occurs only when water within the cells begin to freeze, and this destroys the cell, causing damage. So the muscles in our body can also receive messages from the thermoregulatory center of the brain, the hypothalamus, and that causes shivering. So this increases heat production as respiration is an exothermic reaction in the muscle cells. So basically shivering is even more effective than exercise apparently at producing heat, because the animal remains still. So this means that less heat is lost to the environment through convection. Okay, so apparently there are also different types of shivering, low intensity or high intensity. So during low intensity shivering, animals shiver constantly at a low level for months during cold conditions. But during high intensity shivering, animals shiver violently for a little short time. So both these processes consume energy. However, high intensity shivering uses glucose as a fuel source, and low intensity tends to use fats. Okay, this is the primary reason why some animals can store up food in the winter. the store of food in the winter. And also we have brown adipocytes, right? Brown adipocytes are basically, fat, basically brown fat. Basically they make up the adipose organ together with the white as the adipose tissue. And brown adipose tissue is found in all mammals mostly. Okay, so brown adipocytes are also capable of producing heat via a process called non-shivering thermogenesis. So in this process, triglycerides are burned into heat, thereby increasing body temperature. So that is how our system, the human system, maintains homeostasis. So whatever changes are happening in the outside environment, there is system in our body that is mainly controlled by the hypothalamus to maintain homeostasis in the system. Okay, so till the extreme we see that there are people living in deserts and also in such cold climates, but still they are able to survive because our body, obviously apart, apart from the precautionary measures that we take, there our body has also a very clever system of homeostasis. Okay, so obviously the correct answer is 37 degrees Celsius. The normal body temperature is around 98.6 degree Fahrenheit, 37 degrees Celsius. But there's a range of normal body temperatures, 97 to 99. So I'm pretty sure you are more familiar with the Fahrenheit term in case of, whenever measuring with thermometer. I have seen only Fahrenheit thermometers, but Celsius is more convenient when we talk about things in labs like incubator and all of that. Okay, so basically temperature homeostasis keeps the body temperature within a very narrow range. If temperatures are above this, it can denature enzymes and block certain metabolic pathways. Okay, and temperatures below this can slow down our metabolism and also affect the brain. So heat balance is basically what maintains the body temperature. So there's a balance between the heat production and heat loss. Okay, so heat balance will be like heat production will be almost equivalent to heat lost. And this heat production is called thermogenesis, whereas heat loss is called thermolysis. Okay, so I've already talked about how temperature regulation happens in humans. It is the core body temperature which is maintained by homeostasis, mostly via this hypothalamus. The control of it is under with the hypothalamus, which basically links the endocrine system with the nervous system. Okay, also heat production. We have talked about brown fat. We have talked about chemical thermogenesis. Also heat loss. We have also talked about these scenarios. So we can see these are the mechanisms of heat loss. I've also talked about this radiation, convection, and conduction and evaporation. Basically, sweat glands. If you can see, whenever there's a hot temperature, we tend to sweat a lot. So we have talked about the role of skins also. What is the purpose of sweat? We have talked about the heat exchange via the skin. We have talked about vasoconstriction that happens in cold environment and vasodilation that happens in a very hot environment. So in case of hot environment, arteriolar vasodilation occurs. So the smooth muscle walls of the arterioles relax following increased blood flow through the artery, and this redirects blood into the superficial capillaries in the skin, causing increasing heat loss by convection and conduction. Okay. However, in case of when we are in cold conditions, the arterioles carrying blood to the superficial capillaries under the surface of the skin can shrink or constrict. Okay, so thereby rerouting the blood away from the skin and towards the warmer core of the body. Okay, and this prevents blood from losing heat to the surroundings and also prevents the core temperature dropping further. So this process is called vasoconstriction, and it is impossible to prevent all heat loss from the blood, only to reduce it. So in extremely cold conditions, excessive vasoconstriction can lead to even numbness and pale skin. And as I said, frostbite occurs only when water within the cells begin to freeze. Okay, and this destroys the cell, causing damage as well. So we have talked about temperature regulation by these nervous feedback mechanisms controlled by the hypothalamus. Okay, so we know the feedback system. There will be a receptor that will sense the response to the changes. There will be a control center that will set a range of values. It will evaluate input and send output, and an effector also, which will receive an output from a control center and provide a response. Okay, so the hypothalamus acts like a thermostat. So we have thermostats in countries with very cold temperature, in rooms actually. So hypothalamus, it acts like your own body thermostat. It receives nerve impulses from cutaneous thermoreceptors, and thermoreceptors can be for both cold and heat. Hypothalamus is also has thermoreceptors called central thermoreceptors. So these detect changes in the blood temperature. Okay, This is the structure of the pituitary and the hypothalamus. That is why we say that it's a link between the endocrine and the nervous system. Okay, so what are the neural factors affecting the thermoregulation? There are central thermosensors, there are peripheral thermosensors, and obviously accordingly the hypothalamus will send out information to the sympathetic and the somatic nervous system. Okay, so heat production by shivering, non-shivering thermogenesis in infants, brown fats, blood vessels will internal flow of heat, coat to skin, sweat glands, heat loss by evaporation. All of these signals are sent by the hypothalamus. So, what are the thermoregulatory responses activated by the exposure to cold? Shivering, increase in voluntary activity, increase in TSH secretion, increases catecholamine, vasoconstriction we have already talked about, curling up and all of these. Okay, so basically it's seen that TSH is known to increase during cold temperatures. That has been checked. People have worked on the effect of warm and cool temperatures on the level of TSH. Okay, TSH is basically thyroid stimulating hormone. In case of exposure to heat, we can see there is decreased TSH secretion, vasodilation, sweating, obviously increase in respiration. All of this we have already talked about. Okay, so there are thermo differences in the exposure to cold and heat on the basis of the thermoregulatory responses. So these are a question we can. So what happens in life in hot environment? There are two types, like hot and dry or warm and humid. So hot dry climate is seen in deserts where temperature is above 50 degrees Celsius. Sorry, but they're not really humid. But warm humid will be seen in tropical forests. Temperature will be usually much less than that of deserts, but the humidity will be very much. Okay. So, what are the effects of the acute heat that can be divided into thermoregulatory responses and other effects? Okay, we have also talked about vasodilation, sweating, decreased TSH secretion. Other effects of acute heat can be cellular metabolism, rate of respiration, work of breathing, pulse rate, even urinary volume, dehydration. All of these are effects of acute heat exposure. What are the adverse reaction to heat exposure? Heat syndromes like heat cramps, heat exhaustion, heat stroke. People get heat stroke under very like if they have been standing under sunlight for a very long time. People have been known to get heat strokes as well. Life in cold environment, because of acute long exposure, long-term cold exposure, people can get hypothermia, and obviously there are harmful effects of extreme cold. Shivering, increase in voluntary activity, increased TSH secretion, vasoconstriction, all of these are effects of acute cold exposure in body. So there will be changes in the metabolic response, immunosedative response, hypothermic response due to the long-term cold exposure. Okay. So what is hypothermia? Basically body temperature that will fall below the normal lower limit. Okay. So that is hypothermia. So it can cause frostbite, which occurs at very low temperatures. Surface area freezes, ice crystals formed. Common size can be the ear lobes, your hands, your digits of the hands, your toes, your digits of the feet. So all of these are frostbite indication. So this is a summary of effective mechanisms in temperature regulation. So decrease heat loss, increase heat production. These are stimulated by the cold. Okay, stimulated by heat is increased heat loss, decreased heat production. Okay. So core temperature of 26 degrees Celsius can lead to even death due to brain damage, respiratory insufficiency, cardiac failure, all of the above. So it depends that your core temperature should always be around 37 degrees Celsius. Okay. So it's quite fascinating because in normal, when we talk about room temperature in labs, we basically talk about 25-26 degrees Celsius, correct? At least in India, where we do not use any heaters or anything, we use ACs in lab, correct? Because we have a lot of instruments that need to be around like at least cooler temperatures. So we turn on ACs in here in labs, and the temperature that we keep is around 25-26 degrees Celsius. Cannot believe that our core temperature is 37 degrees Celsius. Can you believe that the temperature you see outside, if the same temperature was inside, like was happening inside your body, it can even lead to death. So 26 degrees Celsius does not seem like a very harmful temperature because that's the normal room temperature here in India. Okay, but when we think if this is a core temperature of your body, it can also lead to death. because it is at least 10 degrees Celsius less than what it is supposed to be. It is 37 degrees Celsius. It can lead to brain damage, respiratory insufficiency, cardiac failure, anything can happen. Okay, so that is fascinating. So moving on to the next question, who coined the term cell? So there are four people listed here: Louis Pasteur, Robert Hooke, Aristotle, and Charles Darwin. Sorry, anybody else? Okay, so Louis Pasteur basically he studied, yeah, he pioneered the study of molecular asymmetry. He discovered the microorganisms cause fermentation and disease. Originated the process of pasteurization, and there were so many beer, wine, silk industries that were saved, and he also apparently developed vaccines against anthrax and rabies. He was the first one to demonstrate that infectious diseases are caused by microbes, disproved the concept of spontaneous generation, and Louis Pasteur is also traditionally considered as the progenitor of modern immunology because of his studies in the late 19th century that popularized popularized the germ theory of disease, and that introduced the hope that all infectious diseases can be prevented by vaccination. Okay, so that was Louis Pasteur. What is the we'll talk about Robert Hooke. Robert Hooke is basically an English physicist who is known for the discovery of the law of elasticity, we know as Hooke's law. Okay, and he was the first use of his word cell in the sense of a basic unit of organisms, describing the microscopic cavities in the cork. And obviously, he is also known for his studies of microscopic fossils, which made him quite famous. Charles Darwin, we know that Charles Darwin is the pioneering figure in the concept of evolution, right? So Darwin's theory of evolutionary selection holds that variation within species occurs randomly, and that the survival or the extinction of each organism is determined by that organism's ability to adapt to its environment. So he's commonly cited as the person who discovered evolution, who proposed the theory of biological evolution by natural selection. Correct? And Aristotle was basically a Greek, one of the greatest philosophers, Greek philosopher, and he has made many contributions to all the fields of philosophy and science. Many things that he has done, honestly, and he apparently distinguished about 500 species of birds, mammals, and all of these in the history of animals and parts of animals. So he has done a lot of Aristotle's zoology and the classification of species was his greatest contribution to the history of biology. The first known attempt to classify animals into groups according to their behavior, and most importantly by the similarities and differences between their physiologies. But the term cell was coined by Robert Hooke, you're correct. Okay. So how it all started basically. So when Romans discovered that they were able to make glass in the 1st century BC, things changed for the better. And we then come to Anton van Leeuwenhoek. Okay, so who was Anton van Leeuwenhoek, and what was his? He was a Dutch microbiologist basically. Okay, and he was universally acknowledged as the father of microbiology. He discovered both, uh, protists and bacteria. He's also known as the father of microscopy. So he was able to discover single cell plants and animals or protists. Okay, so due to his ability to utilize light properly with his microscope, Leeuwenhoek was the first one to give precise shape and size to human blood cells. Okay, and apparently after 2 years, Leeuwenhoek was also able to discover bacteria in water. So he figured he used his 270x magnification compound microscope, and today obviously microscopes magnification range a lot more. So what is the theory basically? A set of principles developed over time by multiple investigations, and the discovery of the cell was made through the invention of the microscope in between this time period. So what is the cell theory? So there are three major parts of the cell theory. All organisms are made of cells. Statement 1. All existing cells are produced by other living cells. Statement 2. And the cell is the most basic unit of life. Okay, so these are the three major parts of the cell theory. So who are the contributors to the cell theory? Robert Hooke, Anton van Leeuwenhoek, Matthias Leiden, Theodor Schwann, and Rudolf Virchow. Each of the above contributors produced evidence through investigations and experiments that led to today's cell theory. So at today's time point, it might seem very simple and all, but you can imagine at that point of time when science was not advanced, when there was technically we were way like you can see we were way more handicapped, like we cannot even begin to compare to today's time. At that point of time, people doing experiments with their rudimentary microscopes and rudimentary knowledge, all of these led to the cell theory that we know today. Correct. So Robert Hooke was an English scientist. He used the compound microscope to observe the cork, and Hooke observed that cork is composed of small hollow compartments. Correct. And these parts prompted Hooke to think of small rooms in a monastery. So he gave them the name cells. Right. We also call cells in any kind of building we can call monastery, or even if we call talk about gel, we can say cells. So that is that basically it made him think of these small rooms, so he gave them the name cells. Now investigate cork through experiment with the compound microscope and came up with the name cells. So he discovered the cell in 1665, which started formulating the cell theory. Right. So these are the Hooke's observations, and this is the Hooke's microscope. Obviously, there will be various YouTube videos and Wikipedia pages that you can search and look into in detail. Anton van Leeuwenhoek was a Dutch person. He studied new approaches for creating lenses to observe clothes, basically. And Leeuwenhoek's microscope was more powerful than Hooke's compound microscope. So, from investigating and experimenting with his microscope, Leeuwenhoek became one of the first scientists to refer to living cells, when he observed an abundant number of single-cell organisms, which he called animalcules, swimming in a drop of pond water. Okay, so this is Leeuwenhoek's observations, Leeuwenhoek's microscope. Then comes Matthias Schleiden. He was a German scientist fascinated with the plant cells. Okay, and Schleiden used the compound microscope and studied plant cells. From investigating and experimenting with plants, projected plant parts are made of cells. So he discussed what he observed with his dear friend, who was obviously Theodor Schwann, who was a German scientist. He studied plant and animal cells and was intrigued by the similarities between the two. So, from investigating and experimenting with plant and animal cells, Schwann was also able to determine that all animals are made up of cells. So, Schwann basically published the first statement of the cell theory: all living things are made of cells and cell products. Then Rudolf Virchow was involved. He, based on his investigations and experiments, he stated that all cells come from pre-existing cells, which is the second part of the cell theory. All existing cells are produced by other living cells. So after Virchow completed his work, cell theory was finally formulated in 1838. So you can imagine at that point of time coming up with these observations and these inferences based on the observations. One second. These inferences based on these observations was not a small deal. It was a huge, huge deal. Okay. So yeah, so the person who coined the term cell will be Robert Hooke, and these people, Robert Hooke, Anton van Leeuwenhoek, Theodor Schwann, Matthias Schleiden, and Rudolf Virchow, all of these people had their contributions to the formulation of the cell theory. Okay, cell theory, which has three statements basically: all organisms are made of cells, all existing cells are produced by other living cells, and the cell is the most basic unit of life. Okay, moving on to the next question for today: which disease causes the most deaths in India, and what kind of organism causes it? Anybody? Anybody would like to try? Sorry, can you repeat? Can you write? I'm not able to hear properly. Maybe you can mute yourself and write. Yeah. Hmm. So anyone else? Sorry. I'm not able to. Hello. So anyone wants to say something, you can write it down also. If you're in a place where there's a lot of noise, you can just write it in the chat box also. I don't know. There's some network issue. I'm not able to hear the voices properly. Anyway, you can write it in the chat box. So if you have said tuberculosis bacteria, that is true. The answer is tuberculosis, and it's not a virus, it's a bacterium. There's the mycobacteria tuberculosis. So we'll talk about tuberculosis and what is the epidemiology of the bacteria, how does it cause so much death in India, and not only in India, also everywhere, but how is it still such a huge scary factor in the world. Okay, so Mycobacterium tuberculosis is a species of the pathogenic bacteria in the family Mycobacteriaceae and the causative agent of tuberculosis. So it was first discovered, I think, by Robert Koch, I think, who gave the Koch's postulates. And M. tuberculosis has an unusual waxy coating on its cell surface, primarily due to the presence of mycolic acid, and this coating makes the cells impervious to gram staining, and as a result M. tuberculosis can appear weakly gram positive. Okay, so acid fast stains such as Zeal Nielsen or fluorescent stains such as Auramine are used to identify the M. tuberculosis with the microscope. The physiology of M. tuberculosis is highly aerobic and requires high levels of oxygen. So primarily a pathogen of the mammalian respiratory system, it infects the lungs, and the most frequently used diagnostic methods for tuberculosis are the tuberculin skin test, Mantof's test, acid fast stain culture, and various types of PCR to check for the particular genes. Okay, so basically it is M. tuberculosis was found in a genetically related complex group of Mycobacterium species. So there are at least 9 members: M. bovis, M. africanum, M. Mycobacterium cannati. So all of these, and it requires oxygen to grow and is non-motile. So M. tuberculosis divides every 18 to 24 hours. That is almost one day here. And this is extremely slow compared to with other bacteria, which tend to have division times measured in minutes. So thus E. coli, Escherichia coli, which is popularly used in labs, can divide roughly every 20 minutes. Okay, M. tuberculosis is small bacillus that can withstand weak disinfectants and can survive in a dry state for weeks. Its initial cell wall is rich in lipids such as mycolic acid and cod factor glycolipids, which is likely responsible for its resistance to desiccation and a key virulence factor. Okay, so M. tuberculosis can be grown in the lab as well. So we talked about M. tuberculosis. So apparently humans are the only known reservoirs of M. tuberculosis. A misconception is that MTB can be spread by shaking hands or making contact with toilet seats or sharing food or drink or sharing toothbrushes. However, the major spread is through air droplets originating from a person who has a disease, either coughing, sneezing, speaking, or singing. So in the lungs, M. tuberculosis is phagocytosed by the alveolar macrophages. Okay, but they are really unable to kill and digest the bacteria. Its cell wall is made up of cod factor like glycolipids that inhibit the fusion of the phagosome with the lysosome, which contains a host of antibacterial factors. Okay, specifically M. tuberculosis blocks the bridging molecule, early endosomal autoantigen 1, and however this blockade does not prevent fusion of vesicles filled with nutrients. In addition, there is a production of isotuberculosinol, which prevents the maturation of the phagosome. So the bacteria also evades macrophage killing by neutralizing reactive nitrogen intermediates. So more recently, it has also been shown that M. tuberculosis can secrete and cover itself in one tuberculosinal adenosine, a special nucleoside that acts as an antacid, allowing it to neutralize pH and induce swelling in the lysosomes. Okay, so then we can see granulomas. M. tuberculosis can actually hide in the granulomas. Granulomas are basically organized aggregates of immune cells, which are a hallmark feature of tuberculosis infection. So granulomas are known to. One second. There's someone here. No, it's not malaria plasmodium. It's tuberculosis. Malaria is not the highest. One second. Does not cause the most deaths in India. It's still tuberculosis. Okay, so tuberculosis have a way to hide in the granulomas. So what are granulomas? Granulomas are basically organized aggregates of human immune cells only, which is basically a hallmark feature of tuberculosis infection. So these granulomas play dual roles during infection. They regulate the immune response and minimize tissue damage, but they can also aid in the expansion of the infection. Okay, so that is how M tuberculosis is can evade the immune system. Also, obviously there are a lot of other strains as well which might not be as harmful as M tuberculosis. So what is tuberculosis? It is caused by a bacterium called Mycobacterium tuberculosis. The bacteria usually attacks the lungs, but TB bacteria can attack any part of the body, such as kidneys, spine, and brain. And TB is preventable and treatable. But not everyone infected with TB bacteria becomes sick. So TB can be active TB, or TB can actually stay in your body as latent. Okay, you won't really have those symptoms that you'll be able to figure out. So two TB related conditions exist: latent TB infection, LTBI, and TB disease, the active TB infection. So if not treated properly, TB disease can be fatal. Okay, so what happens is that TB germs go into the air as aerosol droplets. When someone who's sick with TB disease in the lungs, they cough, sneeze, or speak, and people who are living, working, or spending a lot of time with this person share the same air, correct? So they can breathe in the TB germs and get infected with TB. So you cannot get TB from a person with latent. So there are numerous people having a latent TB infection, but in case of latent TB infection, that person won't really have that bacteria in the spit. So taking spits and doing diagnosis is another way of diagnosing TB infection. So people having active TB will have the TB bacteria in their spit, but people having latent TB will not really have TB bacteria in the spit, MTB. So that is why you cannot get TB from a person with latent TB infection, correct? So TB is not spread by any of these. Okay, so TB germs are spread from person to person by air only. So what are the differences between latent TB infection versus the active TB disease? Okay, so in a person with latent TB infection, it has a small amount of TB germs in his or her body that are alive but inactive. They have no symptoms. So that is why I was saying that there are so many people in the world who probably have LTBI, but they don't even know. They do not feel sick. They cannot spread TB bacteria to others. Okay, so that means they are not contagious. They usually have a skin test or a blood test result indicating TB infection. Okay, they might have a normal chest x-ray. So there might be some lesions in your chest x-ray, but lesions in your chest x-ray can come from various other reasons also. If you have any other lung problem, if you are a chain smoker, that can also cause lesions in your chest. So a person having LTBI can have a normal chest x-ray and a negative sputum smear. Sputum is basically spit. That was what I was saying. So they will not have the bacteria in the spit. But I have heard that people having LTBI might also have lesions in their chest x-ray. So that might be one of the parameters. But then having lesion in your chest x-ray is not the only parameter, because there are other people also who will not have LTBI, but they can still have lesions. As I said, smoking can cause lesions. So there are various other things as well. Okay. So treatment, they need treatment for latent TB infection to prevent the TB disease. But a person with an active TB disease will have a large amount of active TB germs in their body. Okay. Obviously, how will they get to know? Because they'll have so many symptoms. They'll have a bad cough that lasts 3 weeks or longer. They'll have chest pain. They will be coughing of blood or sputum. They will obviously have a lot of weakness or fatigue. There will be weight loss. There will be loss of appetite. You'll stop feeling, stop having the urge to eat. There will be chills, fever, night sweats, etc. Obviously, sick, they will usually feel sick, and they are capable of spreading the TB bacteria to others. Okay, so they can usually have a skin test or blood test result indicating a TB infection. So they will definitely have an abnormal, not definitely, they may have an abnormal chest x-ray, but they should have a positive sputum smear, definitely. So if you take the sputum smear and do culture work in labs, you will be able to see the bacteria growing, correct? And they will obviously need treatment to treat the TB disease. Okay, so what are the risk factors for tuberculosis? Okay, so that is obviously travel to areas where TB is common should be avoided. There will be immune compromise. Okay, living with someone infected with TB is harmful. So what are the TB testing processes? There can be TB blood tests, or TB skin tests. So if either of these tests is positive, further tests are done to confirm a diagnosis of TB disease, like medical examination or chest X-ray. Okay. So another TB skin test is called the Mantoux tuberculin skin test, which requires visit to the healthcare provider. Okay. So in the first visit, apparently a test is placed, and the second visit, the test is read. So there are two different types of tests for TB detection, a blood test and a skin test. So with a blood test, a small amount of your blood is taken for the test. With a TST skin test, a small amount of fluid is placed under the skin of the forearm and requires 2 visits. So if your TB test is positive, it means that you have TB germs, and your doctor may order follow-up chest X-ray and a sputum smear culture to be sure you are not sick with the TB disease. Now, what is the BCG vaccine? BCG is the Bacillus calmette-guerin vaccine. So, BCG is basically a vaccine against TB that is given in countries where TB is very common. I think in India also people get BCG vaccine when they are young. So, it can help prevent infants and small children from getting sick with the TB disease. At best, BCG protects children for up to 5 years. Sometimes it does not protect against TB at all. Okay, so this is the Bacillus calmette-guerin vaccine. I'm pretty sure in India also people take this vaccine as children. I'm not 100% sure, but I've basically heard of this vaccine before also. Okay, and it is basically named after its inventors, Albert Calmette and Camille Guerin. So in countries where tuberculosis is common or leprosy is common, 1 dose is recommended in healthy babies as soon as birth is after birth as possible. Okay, but in areas where tuberculosis is not that common, only children at high risk are typically immunized, while suspected cases of tuberculosis are individually tested and treated. So adults who do not have tuberculosis and have not been previously immunized but are frequently exposed may be immunized as well. Okay. So BCG also has some effectiveness against Buruli ulcer infection and other non-tuberculous mycobacterial infections as well. Additionally, also it is I've heard that it is sometimes used in the treatment of bladder cancer as well. So that is what BCG is all about. Okay, and BCG vaccination. Okay, so the main use of BCG is for vaccination against tuberculosis, right? So BCG vaccine can be administered after birth intradermally, but BCG vaccination can actually cause a false positive Mantoux test. So if you have taken a BCG vaccine and sometime later in your life you are going for a skin test, it might be showing a positive test which will indicate that you have tuberculosis, but probably it might be your BCG vaccine only. Okay, so Mantoux test is not always reliable. Okay, so you need to have other terms of checking if you have tuberculosis, TB or not. So what is the treatment for latent TB infection? So people with latent TB infection should take medicine to prevent the development of the TB disease even though they don't feel sick. And treatment is highly recommended for people who have a higher risk of developing TB disease once they have the TB infection. So this includes people with HIV infection, people who were recently exposed to someone with TB disease, and people with certain medical conditions such as substance use, diabetes, rheumatoid arthritis, all of these. So obviously the thing that causes the most panic is people with latent TB infection won't even understand that they have some illness because they won't be really showing symptoms like that. Correct. But if they are showing symptoms, obviously they need to be treated as soon as possible. And obviously people with other comorbidities, like if they have any other kind of like HIV infection or any other infections like any other disease like diabetes, so those people are obviously very immune compromised. Those people, if you have during the SARS-CoV-2 pandemic as well, if you had seen that people who were young and who were only infected with SARS-CoV-2 were still fine, they were not really showing that much symptoms. But people who were previously ill due to a huge disease or people who had other comorbidities like heart problems, for example, then diabetes, all of these people who were a little obese, all of these people had a lot of trouble, struggling with the pandemic, struggling with the SARS-CoV-2. And even I've heard that even after recovering from COVID, like once they got tested, they turned positive after taking quarantine and everything, they turned negative as well. Even then, there were so many problems. They were feeling fatigued, they were feeling weak, there were other issues in the nervous system as well. So all of these are mostly happening in people with other comorbidities. So that is believable because if your body is already struggling with a disease and you're exposing your body, you have exposed your body to another infection, be it TB, which is caused by a bacteria, or be it COVID, which is caused by a virus. So obviously your body is already struggling. Now you're adding another pathogen to it. So obviously people with comorbidities need to be very careful in the surround whenever they're getting exposed to the outside world, because they need to be extra careful. Okay, so such people should be showing symptoms, and they should be getting treated as fast as possible. Okay, so what are the treatments for the TB, active TB disease? So people with TB disease are contagious because they can pass the TB germs to their family, like how it was in the case for SARS-CoV-2 also, correct? So I do not need to tell everyone what happened during the pandemic. There were lockdowns all over the world. The people were getting quarantined. People could not meet each other for so many days. So because we all know that it was getting transmitted through sneezing or talking, aerosol transmission, all of these were happening. So even if we are going out for extreme emergency cases, we were also taking masks with us. So the pandemic does not need any description. We all have been through the pandemic. So people with TB disease also, they can pass their TB germs to their immediate people who are immediate contacts. Okay, so people with TB disease need to start taking several medicines. Okay, and obviously a doctor will be able to tell TB patients what medicines they need to take based on their previous medical history, and if they have any other comorbidities as well. Okay, and people may undergo direct observation therapy also to make sure that the medicine is being taken correctly and is working effectively. Okay, so for TB patients, there will be some symptoms that will be very easily that can be very easily observed. So say they'll at least have some symptoms, they'll go to the doctor and they'll do tests to figure out what is the problem. But people having latent TB infection is pretty much difficult to diagnose, because they themselves don't show that much symptoms. But at least according to what we have read, latent TB infection containing people might not be able to spread the disease to other people. But during the SARS-CoV-2 pandemic, we knew, right, there were so many people who were not showing any symptoms at all, especially young people who had they had young bodies, and they did not have that much diseases from before. So obviously a young body will be able to cope up with the pathogen much better than a body of an older person, right? So there were so many young people who had the infection, who had the virus, who were incubating the virus in their bodies, but they showed absolutely no symptoms. But that was scary because they were still going out and they could transfer the disease to the other people, correct? And the other people might not be that lucky. The other person might be immunocompromised in some way. That other person might be old. So that was the scary part in case of SARS-CoV-2 pandemic. So COVID-19 disease basically. So as we have talked about tuberculosis, we have talked about how it is caused, and under microscope MTB is identified by its long rod shaped and its structure and its waxy appearance. So these bacteria are single cell organisms. They are able to survive either independently or dependent on other organisms to carry out life functions. So like viruses, bacteria can be beneficial, harmful, or even neutral to the human body. Okay, for example, humans do need several bacteria to carry out bodily functions. Like the gut microbiota, we have so many good bacteria that help us carry out functions. But MTB is often harmful to it's harmful to the human body, and they are often referred to as the TB bacilli. So the thick waxy cell wall allows the TB germ to spread through the air and survive for days outside the body. We already talked about the transmission of TB. It is transmitted through the air when someone infected coughs, sneezes, or anything. So droplets of saliva will contain thousands of TB bacilli, and once inhaled, the droplets will push their way into the lungs, settling in tiny air sacs, alveoli. And TB is not really spread through touch, blood, vaginal fluid, sperm, food, or liquids, shedding intestine, etc. It's completely aerosolic transmission. Okay. So obviously, what are the factors related to the person with TB? The bacillary count or load, its amount of TB in the body, the presence of TB in lungs, pulmonary TB, and obviously there will be accompanied with a cough. So being on effective TB medication, so after 2-3 weeks on an effective treatment regimen, people are usually not infectious anymore, though must continue treatment through cure, and they should obviously wear a mask. There is no second opinion about it. And factors related to the people being exposed to TB through contact, that happens due to closeness and frequency of contact with the index case. Okay, obviously age will also determine. Children and very old people are more likely to develop TB because children they are still developing. Their immune system is still developing. Everything is still developing, and obviously old people, they have more chances of being immunocompromised. They have more chances of getting other diseases. So young people, adults around 18 to 30 will be less prone to showing that much symptoms. Okay, and obviously wearing a mask is also important for them. So obviously environmental factors are also important, ventilation, duration of exposure, all of these. So what is the immune response to TB? If we talk about the body's immune response, the immune system detects foreign particles in the body and triggers an immune response in order to remove them. So in a healthy individual, the immune system will coordinate different cells to act together in order to identify and remove a potentially harmful agent. So when an individual is exposed to TB, the immune system is activated. Okay. So obviously these are the cells of the immune system. We have the APCs, or the antigen presenting cells, basically macrophages and dendritic cells that patrol throughout the body looking for germs. We have the CD4 helper T cells, which basically act as the coordinator of the immune response, instructing other cells to attack specific invading germs. We have the cytotoxic T cells, CD8 T cells, which are involved in cell-to-cell killing. When ordered by CD4 T plus T cells, they seek out and destroy cells that have been infected by a specific germ. We have the B cells, which are immune cells that, when instructed by the CD4 cells, can make antibodies. Correct? And we have the antibodies, which are basically nothing but proteins that attach to the specific germs, marking them for destruction by the immune system or stopping their ability to reproduce. Okay. So what is the immune response to TB? So the antigen presenting cells, or APCs, dendritic cells, are the macrophages. They transport TB to the lymph nodes, acting as the communication and meeting center for the immune system. So in the lymph nodes, if you see, the cells chop up the TB bacilli and present it to the helper CD4 T cell to coordinate the immune response. The cytotoxic T cells are activated to kill cells infected with TB bacilli, and the B cells release antibodies which also target infected cells to kill. Okay. So what is the difference between TB infection and TB disease? So apparently there is a difference between TB infection and TB disease. So it is possible to be infected with TB and not develop the TB disease. So about 10% of the people living with TB infection develop TB disease. People can progress directly to developing active disease without even having a long latent period as well. So that is LTBI, refers to the period latent TB infection period, when the immune system is successful in containing the TB and preventing the progression to disease. So the TB bacilli remains encased in a hard shell called a tubercle. But in case of active TB disease, it refers to the time when the TB is no longer contained by the immune system and it causes the disease. Okay, so they were previously thought to be two different disease states. Now they are thought to be a part of a disease spectrum. So in which latent TB could be early stage active TB with a smaller number of bacteria. Okay, if you see over here, in latent TB lives but doesn't grow in the body, doesn't make a person feel that much sick or have symptoms. So, and can't spread from person to person, can advance to the TB disease. Okay, but in TB disease, if you see, TB is active, it is growing in the body, makes a person feel sick and have symptoms, can spread from one person to the another, it can cause death also if not treated properly. So TB infection can progress to active disease when the body becomes weak, for example from malnutrition, immune suppression, or advanced age. Among people living with HIV and without reliable access to effective HIV treatment, the immune system becomes compromised and more vulnerable to the progression of TB infection into the active TB. Okay, so TB is a very common co-infection of people living with HIV, because when you get HIV, your body becomes immune compromised, correct? And TB infection happens quite easily. So people living with HIV are up to 21x more likely to develop TB disease than people without HIV. Obviously, because people without HIV, their immune system is not compromised unless and until they are infected with some other very serious disease. People living with HIV, their immune system is already very much compromised. Okay, so they develop AIDS, also acquired immunodeficiency syndrome. So obviously it's in the name, immunodeficiency, immunocompromised. So people living with HIV are obviously much more likely to get infected, to develop TB disease. Okay, and obviously young children are up to 10x more likely to develop TB, and they also tend to develop more severe forms of TB. Obviously, because in young children the immune system is still developing. Okay. So active TB disease affects the lungs, pulmonary TB, or other parts of the body, extrapulmonary TB as well. Pulmonary TB, the lungs, the TB that affects the lungs is the most common form of TB disease. Extrapulmonary TB can occur in all populations affected by TB, but is most common among young children and people with HIV. Okay, so it can take place in multiple organs in people with HIV as well. Okay, so individuals can have pulmonary TB, extrapulmonary TB, or they can have both as well. Okay, so what is the immune response in children? A functional immune response system takes many years to develop in a child. That is why children are so much more prone to TB. So when exposed to TB, young children less than 5 years cannot usually mount a robust immune response. For this reason, they are more likely to develop severe forms of TB, including TB outside the lungs, extrapulmonary TB. Children who are immunocompromised due to HIV, malnutrition, or other sicknesses are also at risk for developing more severe forms of TB. As children age and the immune systems develop, they can better control TB when exposed. If they get sick, they tend to have a disease that is more like adults. Adolescents ages 10 to 18 years are more likely to develop TB. This may be due to the impact of hormones or puberty on the immune response to TB and increased social activity. Obviously, so as you grow older, you'll have more social activity around you. So you might get exposed to TB infection more. Okay, TB can happen in children. It can actually cause disease any part of the body. Okay, but the most troublesome ones are TB meningitis. Okay, it's the TB in brain nervous system. Imagine how serious it can be. The miliary or disseminated TB throughout the body. The Pulse disease, TB in the bones and spines. 70 to 80% of children have TB in the chest and lungs, which is a pulmonary TB, most common form of TB. Children can become sick from a smaller number of TB germs, and children are more likely than adults to develop TB outside of the lungs as well. So in like compared to adults, children are more prone to TB outside of the lungs, like extrapulmonary TB as well. So what is drug sensitive and drug resistant TB? So drug resistance or DRTB means a strain of MTB that has mutated or changed in a way that helps it evade or resist being killed by specific drugs. So resistance to TB drugs can be naturally occurring, wild type, or develop over time as a result of inadequate or irregular TB drug exposures. Okay. So like incorrect prescription, poor quality drugs, drug shortages resulting in treatment interruption or lack of adherence to the treatment. Okay, so that is why, an AMR, if you have heard of AMR, antimicrobial resistance, is such a huge problem in today's time, especially because people don't complete the antibiotic dose. Okay, so when you are sick, it is always recommended that you consult a practicing physician before you take antibiotics. You do not take antibiotics for minor colds or anything. You should always consult a qualified person who will know your medical history, who will be able to prescribe you antibiotics properly. So what people do is that people get fever, little bit of fever or cold, and they take antibiotics just like that. Okay, and what they do wrong is that they start taking the antibiotic. After 2 days, they're done with it. Okay, they don't take it anymore. They're like, oh, my symptoms have subsided, fever is not back, so I'll stop taking the antibiotics. That is such a wrong step, because you have started the antibiotic dosage and you're not completing the dose. So what happens, the bacteria in your body, which have not, your body has not eradicated the bacteria, there are still bacteria in your body. Obviously, because of the drug or the antibiotic, their functioning might have gone less, but they are still in your body. So what happens, the antibiotic is in your body, the bacteria is not entirely eradicated, but you have stopped taking the antibiotic. So now the bacteria has a chance to evolve so as to evade the antibiotic. It will find out a way to mutate. It's constantly mutating. It will find out a way to mutate so that it doesn't get affected by the drug anymore. So after that, even if you take drugs, the bacteria, it won't harm the bacteria, because now the bacteria has become drug resistant. Okay, so that's why it is so much recommended that you do not take antibiotics without consulting a physician, a qualified physician. And obviously, if they give you antibiotics, they will probably tell you, but even if they don't, it's always recommended that you complete the dose. So when you complete the dose, what happens? You make sure that all the bacteria in your body has been eradicated by the continuous intake of antibiotic, right? If you stop in the middle, your bacteria will have a chance to evolve, mutate, and become resistant to the bacteria. So that has what led to these resistant drug resistant TB and all of that. Okay, so poor quality drugs resulting in inadequate drug levels, incorrect prescription by healthcare provider, drug shortages resulting in treatment interruption, lack of adherence to the treatment, very important. All these are man-made problems that have led to the drug resistance of so many bacteria around us. Okay, so most drug resistant TB is transmitted rather than developed. Okay, so it's transmitted rather than you develop it yourself. And treatment for DRTB is longer, obviously more expensive and harder to tolerate than treatment for drug sensitive. That is, isn't that obvious? Because a drug sensitive TB can be easily countered, because you'll take drugs, you'll complete the dose and you'll be fine. But if your body, if the bacteria in your body is not reacting, is not being subdued by the drug or the antibiotic that you're taking, it's nothing is happening, then it's much more expensive and obviously very hard to tolerate, and obviously it's a it's a bunch of new problems for the patient. Okay, so drug resistant TB is also generally separated into 4 groups defined by the medicines to which TB bacteria are resistant. Okay, so these are the defining drug resistant TB. Drug sensitive TB will be sensitive to these these antibiotics, these drugs, rifampicin, isoniazid, fluoroquinolone, aminoglycoside. For multi-drug resistant TB, they are resistant to rifampicin and isoniazid. For XDR TB and pre-XDR TB, they are basically resistant to all of these. XDR is extensively drug resistant TB. It is a rare type of multi-drug resistant TB that is resistant to isoniazid, rifampicin, fluoroquinolone, all of these. Okay, so that is XDR TB. And what is pre-XDR TB? Pre-XDR TB is basically caused by an organism that is resistant to all of these. Okay, it's pretty much similar. So, so research is going on, obviously, to find out drugs that can target and eradicate the XDR TB as well. So obviously the global statistics is important. It is estimated that 1/4 of the world is infected with latent TB. Okay? And there were an estimated 10 million new cases of TB in 2019, among which 500,000 had drug resistant TB. So it's a very huge number and it is very scary. Okay, so TB and COVID-19. So COVID-19 caused by SARS-CoV-2, the respiratory pathogen that emerged in late 2019, as we all know, and has caused sickness in millions of people. And symptoms of COVID-19 and TB may overlap, because we know fever, cough and all of these. And persons being tested for COVID-19 should also be tested for TB. Okay, there have been instances where there have been co-infections of both seen in people. Persons with current TB or a history of TB may be at increased risk of poor outcomes if they become sick with COVID-19. So they should take extra care to practice mask wearing, social distancing and everything. And the global COVID-19 pandemic has put at risk many of the strides made addressing in TB on a global level. Okay, so WHO has model estimates that 25% drop in the number of people diagnosed and treated for TB over a 3-month period will result in 200,000 excess TB deaths. Okay, so COVID-19 and TB co-infections have also become quite common during the COVID-19 pandemic. So what are the main points, the key takeaways? Tuberculosis is a disease caused by the bacterium Mycobacterium tuberculosis. It spreads through saliva droplets in the air when a person sick with pulmonary TB coughs, sneezes. There is a spectrum of TB disease, including active TB disease when the presence of MTB causes disease, or latent TB disease, LTBI, when MTB is present without the disease. Drug resistance is a result of inadequate or irregular TB drug exposures, which is on the rise and evolving with increased use of new and repurposed TB medicines. TB is a common co-infection and the number 1 killer of people living with HIV, another popular co-infection, and coordinated efforts are needed to address the TB in the era of COVID-19. Okay. Coming to the next question for today. What substance comprises the genetic material of the SARS-CoV-2 virus? Anybody? RNA. So do you know if it's positive stranded, or is it what is the number of strands of the RNA? You know the genetic material of SARS-CoV-2? Sorry. Okay. Yes. So yeah. So SARS-CoV-2 is a single-stranded RNA material. The genetic material for SARS-CoV-2 is ribonucleic acid. Okay. So moving on, RNA. So obviously we are not COVID-19 or SARS-CoV-2 does not need any introduction. Obviously. So there there are 7 human coronaviruses, 4 normal and 3 novel. Okay, we'll be talking mostly about the COVID-19, whatever caused the COVID-19, which comes under the beta coronavirus. So the coronavirus structure is a medium-sized virus size but largest mRNA genome. So it's an enveloped positive stranded single-stranded RNA, and the mRNA is encased in the nucleocapsid. Okay, there's a lipid bilayer as well, and soap is known to disrupt this lipid bilayer, which is why during COVID-19 pandemic we were advised to wash our hands with soap for 20 to 30 seconds. If you remember, we every time we come from outside, we were encouraged to wash our hands with soap to disrupt. Even if we have the virus particles in our hand, washing with soap will probably disrupt this lipid bilayer. Okay, and why is it named corona? Because of the crowns, the spike protein on the COVID of the virus, basically gives it the name corona. Okay, so this is a glycoprotein spike aptamer, and the spikes allow it to attach to human cell receptors in the upper or the lower airway. So the coronavirus genome basically encodes 4 or 5 structural proteins. The spikes, which is obviously the most studied one because it's on the outside, it is what gives it a crown-like appearance. The corona name comes from that, and obviously it mediates receptor binding. So the spikes can attach to the ACE2 receptor, okay, on human cells, and that can mediate receptor binding. Then we have membrane protein that assists in viral assembly. We have nucleocapsid protein that helps in regulation of viral RNA synthesis, and it may interact with the M protein during virus budding. We have E or small envelope protein that function is necessary but still is being studied. It's not fully understood. And we have the HE, hemagglutinin esterase glycoprotein, the beta coronavirus OC43 and HQ1 only. So it enhances uptake into the mucosal cells. Okay, so normal human coronaviruses cause 5 to 10% of the common colds. Okay, with outbreaks to 30% of common cold. These 4 predominantly attach to receptors in the upper airway. Receptors can be aminopeptidase N, dipeptidyl peptidase 4, and seasonality unpredictable. So generally winter but persists year round. Different pattern in tropics and temperate regions. The novel coronavirus, predominantly in lower respiratory tract, those are SARS-CoV. I think the SARS-CoV was detected in 2003. SARS-CoV-2 was detected in 2019, which led to the recent pandemic. Okay, so obviously there are other lower respiratory tract infections, such as viral pneumonia caused by influenza, adenovirus, all of these typical bacteria such as Haemophilus influenzae and all of these. So the SARS, yes, as I said, it was in 2003. MERS was not really contained, and there was a lot of nosocomial spread. So nosocomial, does anybody know the meaning of nosocomial? So nosocomial infection is basically infections that basically healthcare associated infections. So they are infections acquired during the process of receiving healthcare that was not present during the time of admission. So for example, if you have a particular problem and you get yourself admitted to a hospital, you might get an infection while you're in the hospital. Okay, so it's the hospital acquired infection or healthcare associated infection. Okay, so patients and healthcare professionals can bring germs inside the hospitals and pass them to each other. Sometimes people carry these germs even without feeling sick or showing any symptoms, which means they can spread them to others without knowing. Okay, and if you're already in the hospital for any kind of disease or surgery or another illness, obviously immune system is too weak to fight off these germs. So that those are called nosocomial infections. So how does the novel coronavirus attach to the human cells? As I said before, the spike protein attaches to the ACE2 receptor. Okay, you can see over here. Okay, it binds to the ACE2 receptor, and then it is taken inside the system. So what is the origin? So people say the origin is from a bat to a mammal. So that is why it's called a, if you know the term, it's called a zoonotic virus. Okay. So what is a zoonotic virus? A zoonotic virus is a disease or infection that can be transmitted naturally from animals to humans, or vertebrate animals to humans, or from humans to vertebrate animals. Okay. So SARS-CoV-2 is apparently a zoonotic virus. Okay, because COVID-19 caused by the SARS-CoV-2 was most likely originated in bats and transmitted to humans through a possible intermediate host. Okay, so that is also SARS-CoV-2 transmission. We obviously know about the incubation period, which was why we were asked to quarantine for 7 to 14 days before, and then it got reduced to 7 days eventually. The transmission occurs due to respiratory droplets, hand to mucous membrane contact, anything. It is also apparently viable for 3 days on solids, 24 hours on cardboard and everything. Okay, so all of these were the methods of transmission. So droplet borne route transmitted by medium or large droplets or short range airborne droplets. So all of these were the illustration. This is the illustration of different transmission routes. Small droplets, sometimes called aerosols, are responsible for the short-range airborne route, long-range airborne route, and indirect contact route. Large droplets are responsible for the direct spray route and the indirect contact route. Okay. So the answer was RNA. Coming to the next question for today. Okay, so the answer came first anyway. So most enzymes are proteins. So that was the question. But can you name another molecule inside the body which can also act as enzymes apart from proteins? Anybody? Anything that can act as an enzyme apart from proteins. Sorry? DNA. No, DNA cannot really act as enzymes. But you're in the right path. It is a nucleic acid. Yeah. RNA. So, like proteins, RNA molecules can also be enzymes, and they are in such case, those are called ribozymes. Okay, so ribozyme is a ribonucleic acid RNA enzyme that catalyzes a chemical reaction. So, the ribozyme can catalyze specific reactions in a similar way to that of protein enzymes. Okay, so catalytic RNA ribozymes are found in the ribosome also, where they join amino acids together to form protein chains. And ribozymes as catalytic RNA molecules were first identified in the 1980s. Okay, so they have the intrinsic ability to break and form covalent bonds in RNA molecules. So in many ways, they can be compared to the protein enzymes which catalyze cleavage of peptide bonds in other proteins or peptides. Okay. So obviously the answer here will be proteins. Most enzymes are proteins. Okay, so if we talk about the thermodynamics aspect, when the reaction will occur, but if we talk about the kinetics aspect, we think about when the reaction will occur, how much time it will take, etc. So what governs when a reaction will occur? Okay, so oxidation of carbohydrates, polymers to carbon dioxide and water is favorable, but when will it happen? Gasoline burning to carbon dioxide, CO2 and water is favorable, but when will it happen? So for a reaction to occur, we need to destabilize the existing state, correct? And we need something that will basically decrease the activation energy, correct? So what does the activation energy represent for a reaction to occur? So reactants must find each other, they must meet in proper orientation, and they must be hit with a sufficient force, okay? So needs of typical chemical reactions need large number of molecular collisions. We need the collisions to be violent enough to break pre-existing bonds. We need high concentration to find each other at a significant rate. Okay, and so this is the, if you look at the energy profile for an hexagonic reaction, you have the reactants, you have the products, and this is the activation energy. Okay, so what is the role of catalysts? Catalysts basically lower the activation energy barrier. They remain unchanged even after the reaction, but they lower the activation barrier. Okay, so if you see over here, this molecules with the average energy, and many molecules have enough energy to undergo enzyme catalyzed chemical reaction, but almost no molecules have the very high energy needed to undergo an uncatalyzed chemical reaction. Okay, so that is why we need a catalyst. Okay, so enzymes make reactions easier to occur at reasonable temperature by lowering the activation energy of the reaction. Enzymes are biological catalysts, nothing else. So as we know, catalysts lower the activation energy of the reaction. Enzymes act as biological catalysts and lower the activation energy of the enzyme. Activation energy of the enzyme. Okay, 1 second. Maybe. Um, okay. Okay. So this is the activation energy of the reaction, and we need the activation energy to be lowered for the reaction to take place properly. So as we know, catalysts are not consumed in the process, they remain unchanged, but they promote a specific reaction. Okay, so they promote, does not alter what would normally occur thermodynamically, and they also promote only one reaction only between specific reactants to give specific products, and they can be regenerated in the process. And enzymes are biological catalysts, they're usually proteins, sometimes RNA or RNA protein complexes. So enzymes work as catalysts by providing an easy path to the same point, correct? So how do enzymes do it? Enzymes have binding affinity for the reactants such as substrates. So if this is an enzyme, uh, it will have a binding affinity for a specific substrate, correct? And it will bring substrates in close proximity. And there will be stabilized interactions. They will have a very specific 3D shape with a specific arrangement of functional groups. So enzymes will act in a specific pattern. Also, it will bind only specific things, bind them only in a specific 3D orientation. So specificity is basically the key to enzyme action. If you have read about lock and key model, so if you see the lock and key model illustrates that only. Okay, so enzymes will orient the substrates always in productive orientation. Okay, so if you see this is a productive collision. Okay, collision is happening and they just fit into each other, but this is a non-productive collision, nothing is happening over here. So only the enzymes cause bond strain. Obviously they will destabilize the existing bonds, okay, of the reactants. There will be physical strain, there can be chemical strain. Okay, there is an active site in the enzyme. So the active site of the enzyme is the region on the enzyme where the substrate will bind. If this is a substrate, this is the enzyme. If this, you can call this the active site. The substrate will come and bind over here. Okay, so induced fit of a substrate. This is how the substrate will bind and the enzyme will be induced. So this will call the induced fit of the substrate. Correct. So the induced fit model basically states that a substrate binds to an active site and both change their shape slightly, creating an ideal fit for catalysis. Okay, so enzymes promote chemical reactions by bringing substrates together in an optimal orientation, thus creating an ideal chemical environment for the reaction to occur. Okay, so this is the Fisher lock and key model where this is the active site, this is the enzyme, this is the substrate. So basically they fit perfectly together like a lock and key, and then the products are formed which are then released. For the Koshland's induced feed model, so the substrate comes and there is a little bit of change in both changing shape in both the molecules so that they're exact fit for each other. Okay, so this is a catalytic cycle of an enzyme. The substrates enter the active site, the enzyme changes shape, so its active site embraces the substrate. This is the induced fit. Then the substrates held in the active site by weak interactions such as hydrogen bonds and ionic bonds. Then the active site, the R groups of the amino acids can lower the activation energy barrier or EA and accelerate the reaction by acting as a template for substrate orientation, by stressing the substrates and stabilizing the transition state, by providing a favorable microenvironment, or by participating directly in the catalytic reaction. Okay. Then the substrates are converted into products and the products are released. Okay. The active site is now available for two new substrate molecules. This is the catalytic cycle of an enzyme. So you see the enzyme does not get consumed in the process. The enzyme does not get taken up in the process. That is what the primary criteria of a catalyst is. So what are cofactors? Cofactors can be non-polypeptide things at the active site that help enzymes do their job. So they are non-protein enzyme helpers such as ions. Cofactors can be Zn2 plus ions. or they can be coenzymes like organic cofactors. Okay, so enzymes partake in reactions but are not consumed in them, as I said already. So we have an enzyme, the active site substrate will act, there will be enzyme substrate complex, enzyme product complex, and the enzyme will ultimately release the product. Okay, so if you see over here, if you can think of the particular enzyme, if you see the mechanism of action of many enzymes such as chymotrypsin, trypsin, all of these, you can see how the mechanism of action takes place and which are the particular amino acids in the active site that are responsible for that. Okay, so let's obviously the enzyme can catalyze a reaction. So if you see, this is the Michaelis-Menten curve. This is a substrate concentration, and we have the rate or velocity. This is a V max. Okay, so that is what we get the V max from, and so this will be the half V max. Okay, and this will be the half V max. Can be extrapolated here, and that will be the substrate affinity or Km. So the lower the Km, the better the enzyme recognizes substrate. Finds it at low concentration. So the higher the Vmax, the more substrate an enzyme can process per minute. Okay. So things that affect protein structure often affect enzyme activity. Obviously, take changes in enzymes are showing optimal activity at particular temperature ranges, at particular pH ranges. So these, for example, this is optimal temperature for the typical human enzyme. This is optimal temperature of a thermophilic heat tolerant bacteria. So for example, this is a pH optimum for pepsin. This is the pH optimum for trypsin. Right? pH optimum for pepsin is quite low because pepsin is an enzyme that is has a very low optimum pH. At an optimal pH of around 2 allows pepsin to operate because its natural acid, because the natural environment is acidic. Okay, so the pepsin cannot really work at neutral pH. Pepsin will get denatured at neutral pH. Its natural environment is acidic, so that is why this is the pH optimum for pepsin. Okay, enzyme regulation can be activity controlled or continually adjusted. So what are the principal ways of regulating enzymes? By competitive inhibition, allosteric inhibition, and by any kind of covalent modifications such as phosphorylation. So what happens in competitive inhibition? There are competitive inhibitors that are added, which basically compete with the substrate to bind to the active site unproductively. So once the competitive inhibitors are given in excess, they can bind to the active site, and that basically stops the substrate from binding to the active site. They are binding to the same site. So there is a competition. That's why it is competitive inhibitor. So if you see, this active site of the enzyme, if the substrate is added, it will bind to the enzyme and the product will form. But if we add a competitive inhibitor, the competitive inhibitor will bind to the active site and will consume the active site. Okay, so we'll basically block the active site. So the enzyme cannot bind to the substrate anymore. So there is competition, that is binding of the inhibitor prevents the substrate binding. So this is competitive inhibition. In allosteric inhibition happens at another site from the active site. So basically what happens, this is the enzyme, this is the active site of the enzyme. So substrate can come and bind over here. So what happens if we add an allosteric inhibitor? For example, here it is a negative allosteric inhibitor regulator. So if we add an allosteric inhibitor, it changes the structure of the enzyme's active site in such a way that it does not have any affinity to the substrate anymore. So see, it is a proper lock and key model if you see the over here. But once the allosteric inhibitor, which is a negative allosteric regulator, binds to the enzyme, the active site changes in such a way that it does not have an affinity towards the substrate anymore. Okay, if you see over here also, this is the active site, this is the allosteric site. So the substrate can bind to it and products can be formed. But if you add an allosteric inhibitor, it will change the active site and little or no product formation can occur. So an enzyme subject to allosteric inhibition is active in the uncomplex form, which has a high affinity for its substrate. So binding of an allosteric inhibitor stabilizes the enzyme in its low affinity form, resulting in little or no activity. What are positive allosteric regulators? So in case of positive allosteric regulators, for example, if you see the enzyme is not exactly a perfect fit for the substrate, correct? So if we have an positive allosteric regulator, we will not call this an inhibitor because it's not really inhibiting the process. If we have a positive allosteric regulator, it will bind to another site, the allosteric site, and it will change the active site of the enzyme in such a way that now it becomes a perfect fit for the substrate. Okay, so positive allosteric regulators are positive because they help the enzyme work better because it promotes, stabilizes, an active conformation. Okay, so this is what we call a positive. So before, the active site was not really compatible with the substrate, but after the allosteric positive allosteric regulation has happened, binding of the allosteric activator, which is a green in color, shown in green in color over here, it stabilizes the enzyme in its high affinity form, resulting in the enzyme activity. Okay, so allosteric regulators change the shape conformation of the enzyme. So that is the main purpose of allosteric regulators. So it can either be an allosteric activator or it can be an allosteric inhibitor. So for example, here if we see, it's an allosteric activator. So it activates the active form. Okay, it stabilizes the active form, sorry. But in case of an allosteric inhibitor, it basically stabilizes the inactive form. Okay, that is what is the role of allosteric regulators. Another frequent regulatory modification is the phosphorylation of enzymes. So this is an enzyme which is phosphorylated to this structure using protein kinase. And if you want to remove the phosphate group, we have to treat it with protein phosphatase. So kinase and phosphatase, these two enzymes have opposite functions. Okay, so if you read the chapter on cell signaling from Bruce Alberts, you see that there are so many signaling molecules that are turned on and off, like their functions are turned on and off by the means of phosphorylation. So for example, this is a particular enzyme, its function is turned off now. So on addition of the kinase, it will have a phosphate group which will basically turn on the function of this molecule. Similarly, with phosphatase, we can remove the phosphate molecule as well. So it's very relevant for signaling molecules. Okay. So phosphorylase kinase, we have also talked about this. So in summary, we know that the enzymes are catalysts. They lower the activation energy. There we talked about some of the mechanism of actions. We have talked about enzyme kinetics and Michaelis Menten. We have talked about the regulation of enzyme activity, competitive allosteric phosphorylation, etc. Okay. So these are enzymes. We have already talked about that they are the key to life. Okay. We have talked about what is an enzyme, active site. So it's a globular, any protein which functions. It can be RNA also, but mostly since we are talking about proteins over here, let's stick to proteins which function as biological catalysts, speeding up reaction rate by lowering the activation energy without being affected by the reaction it catalyzes. Okay. So enzymes are protein in nature, can be globular protein, ribozymes are RNA molecules with enzymatic activity. Okay, so if someone tells you only proteins can be enzymes, that is a wrong statement. RNA molecules can also be enzymes. Till date, to whatever level our knowledge has reached, RNA molecules can also have enzymatic activity. Okay, catalytic behavior of any enzyme depends upon its primary, secondary, tertiary, or quaternary structure. We have already talked about the different levels of structure of protein in one of the previous class. Enzymes of digestive tract and those found in blood are present in inactive form called zymogen or proenzymes, and they are then converted into their active form when needed. Okay, so what is active site? Enzymes are composed of long chains of amino acids that are folded into very specific three-dimensional shape which contains an active site, correct? So an active site is a region on the surface of an enzyme through which substrates will bind and catalyze a chemical reaction. So enzymes are highly specific for the type of reaction they catalyze and for their substrate. Enzymes with highest specificity and accuracy are involved in copying and expression of genome. So these enzymes have proofreading functions. So we can talk about DNA polymerase, which is an enzyme. Some enzymes require non-protein molecules for their activity. So enzymes are conjugated proteins known as holoenzymes. So holoenzyme will be apoenzyme plus cofactor of the coenzyme. Cofactor can be your ions also, like Zn2 plus as we discussed, and it can be coenzymes like organic molecules as well. Enzyme without non-protein part is inactive and is called an apoenzyme. So cofactors can be either inorganic or organic compounds. Okay. So what is the mechanism of enzyme action? The enzymatic reactions will take place by binding of the substrate with the active site of the enzyme molecule by several weak bonds. We will form an enzyme substrate complex, which is the first step, and then the ES complex will be subsequently converted to free enzyme and the product. Okay. We have also talked about the lock and key model and the induced fit model. So we have talked about the induced fit model, that the substrates enter the active site, enzyme changes the shape, so the active site now embraces or properly fits the substrate. That is the induced fit. The substrates held in active site by weak interactions such as hydrogen bonds and ionic bonds. Then the active site, which are the R groups of its amino acid, can lower the activation energy and speed up a reaction by acting as a template for substrate orientation, stressing the substrates and stabilizing the transition state, or providing a favorable microenvironment, participating directly in the catalytic reaction, etc. Then substrates are converted into products and the products are released, and then the active site is again available for the new substrate molecules. The enzyme remains unchanged. Okay, we have talked about the progress of reaction. This is without enzyme, you can see the activation energy is so high, but with the biological catalyst, the activation energy has been lowered. Okay, so obviously there are various factors affecting reaction velocity. Temperature. So every enzyme will have an optimum temperature. Every enzyme will be able to work at an optimal pH. The substrate concentration, enzyme concentration, products of the reaction, all of these, the presence of any activator or inhibitor, all of these are very important factors in affecting reaction velocity. Okay. So what can be the effect of temperature? So there will be an optimum temperature for every protein. So if you can see, if for example, for a particular enzyme, if you plot the x-axis with temperature and y-axis will be reaction velocity. If you see, so this particular enzyme has 40 to 45 degrees Celsius as its optimum temperature because it shows the highest reaction rate or reaction velocity at this particular temperature. Okay, effect of pH can also be shown by graphs over here. So you can see for pepsin is an enzyme which is present in naturally acidic environments. So its optimum activity is around 1.5 to 2 pH. But trypsin works better in at 7 to 8 neutral pH conditions. If you hope that pepsin will act at such a condition, it will not. Pepsin will denature in neutral pH conditions. So every enzyme has a optimal pH range in which it will function with the greatest reaction velocity. Okay. So the rate of the reaction or velocity will be directly proportionate will be to the enzyme concentration when sufficient substrate is present. When substrate is not the limiting reagent, the more you increase the enzyme, there will be more reaction velocity or rate. Okay, but if there is a lot of product that accumulates in a reaction, it might cause a negative feedback loop inhibiting the enzyme activity. Okay, effect of substrate concentration that can be seen with the Michaelis-Menten reaction. Okay, so we have already talked about the Michaelis-Menten reaction. Obviously, enzyme kinetics can be studied. Basically, an amount of substrate present is one of the key factors affecting the rate of reaction catalyzed by an enzyme in vitro. Effect of substrate concentration on reaction velocity can be studied by the means of Michaelis-Menten. So, on the x-axis, we have substrate concentration. On the y-axis, we have initial velocity. Okay, this is the Michaelis-Menten kinetics. So the Michaelis-Menten equation comes at V0, V naught equal to V max S by Km plus S. Okay, so this mathematical equation that defines the quantitative relationship between the rate of an enzyme reaction and the substrate concentration is Michaelis-Menten. So if you make a reciprocal of this equation, you will find the Line-Weaver-Burk model. Okay. That is a double reciprocal. Both reciprocals or linear representation is more accurate and convenient for determining Vmax and Km. So this equation is obtained by taking the reciprocal on both the sides. So 1 by S and 1 by V naught. So this will give you the Line-Weaver-Burk or the double reciprocal plot. We have talked about enzyme inhibition as well. Types of enzyme inhibition can be reversible or irreversible. Reversible inhibitors can be competitive, non-competitive, uncompetitive. Competitive we have already talked about. Substrate will attach and the inhibitor also attaches in the same place. So it will basically compete with the substrate for attaching to the enzyme. For non-competitive, the inhibitor attaches somewhere else. It is not exactly competing with the substrate. And uncompetitive, it binds only to the enzyme substrate complex. It does not have a capacity to bind to the free enzyme. Okay, and so that is why it is not overcome by the increasing substrate concentration. So this is what we mean by uncompetitive. It is binding to the ES complex. Okay. So we have talked about the different types of enzyme inhibition. Okay, so we already know what are proteins. They are made up of chains of amino acids. They are found in every cell in the body. So obviously the role of proteins can act as structural components of the cell. They can act as enzymatic components. They can act as they have many different functions. So the peptide, the acid group of one amino acid joins with the amine group of the second amino acid, it will release H2O. There will be a condensation reaction, and that will basically form the peptide bond. So a peptide bond forms by the condensation when the acid group and amino group of two different amino acids basically join and release a molecule of water. This will be a peptide bond. So when peptide bonds are broken by hydrolysis, the hydroxyl group and the hydrogen from water are added. We have also talked about the essential and non-essential amino acids. So essential amino acids are something that your body cannot produce. So these are very much essential in your diet. Non-essential can be produced in the body, so they are non-essential in your diet. So you have talked about the 20 amino acids. You have also talked about the different levels of structure of the amino acids. Structure of the proteins. There can be primary structure, which is basically the primary sequence of amino acids. Secondary structure, which can be alpha helix, beta sheets, turns, loops, etc. Tertiary structure is basically how the polypeptide chain basically folds upon each other. Basically a 3D structure. Quaternary structure will be how the different polypeptides making the different subunits come together to form a particular protein. That is the quaternary structure. Denaturation can happen, that is altering the protein's shape and structure so that it becomes non-functional, or it goes down in its efficacy. Can be done by heat, acids, bases, salts, etc. Okay, so denaturation cannot change the primary structure as such. It will change the folding, and if the folding of a protein is not proper, its functions will not be proper. So that is what denaturing does. So normal protein. So with denaturation, the primary sequence is not affected. If you see, sequence is there, but the folding has disrupted. So without the proper folding, the structural function of the protein or the enzymatic function of the protein, the active sites which were supposed to be there because of the folding of the structure will not be present properly, and your denatured protein will be non-functional. Okay. So that is what we have talked in this. So in this question, the question was, which of the following can act as enzymes? So obviously proteins are very popular as their role as enzymatic catalysts, biological catalysts, enzymes. Apart from protein, RNA or ribonucleic acid also they also can function as catalysts. Okay. So today we'll stop with this question. Does anybody have any queries related to all the questions that we discussed today? Or if you don't have any questions, you can write it down also. Anybody? Okay, if we don't have any question, maybe we can conclude today's session. Okay, and we'll meet again next Monday. Okay, thank you everyone.
Thank you. Thank you.
Okay, we'll conclude today's session.
Bye.
Thank you.